Well, I think we'd best start now. I suspect that we've got most people entered. I'm going to say thank you to Dana and all for inviting us, and that Dana says that she regrets that she can't join us today. So I'll be introducing me. I am a Canadian Blood Services scientist by primary appointment. I've been with the Center for Blood Research since inception, and I'm a proud member of the Department of Pathology and Laboratory Medicine here at UBC. And I'd like to thank you all for joining us to bravely learn about that complex clotting pathway, which is what my laboratory toils over, well, day and night. An important take-home message that I would like to stress today is that clotting enzymes do so much more than merely control bleeding. And well, you can Google that. Pick any of your favorite physiological or pathological process, and I bet you're going to get some hits if you link it to coagulation or clotting. Today, we're going to tell you about two processes, clot busting and virus infection, and how they are linking to coagulation. And we're going to play a pretty significant tag team. We're going to start out with Alexandra Witt, who didn't really know she signed up to give a talk less than 2 months since she started with us as a summer student, but she is going to be starting as a graduate student in September. So this is good practice for her. We're going to be starting, seconding her with Scott Meixner, who is a senior research assistant, long-term vet in my lab for about 16 years. They're both going to be talking about clotting factor 10 and how it participates in the complete opposite role as a clot busting protein. Then we're going to shift gears and talk about clotting factors in the context of viruses. And that is going to be led first with Dr. Michael Sutherland, who's been with me for over a quarter of a century as a research associate. And he's also an adjunct professor here at UBC. He's going to tell us about how this very important enzyme called tenase, and that is aptly named because it activates clotting factor 10, how it enhances virus infection. He's going to be focusing on the cold sore virus, oral herpes, HSV1. He's then going to hand the baton over to Henry West, who is a graduate student, who's going to talk more about that tenase and how it exists on the surface of a whole bunch of viruses. And he's going to then shift gears to SARS-CoV-2 and tell us about how SARS-CoV-2 induces that tenase on the surface of cells. Then we're going to talk more about SARS-CoV-2 and Alex Leatherdale, a postdoc who was co-supervised by our favorite Dr. Ed Conway. He's going to tell us about how SARS-CoV-2 induces tenase antigen on cells in line with Henry West's cellular tenase. And he's also going to tell us about SARS-CoV-2 triggering complement. And that is partly in line with how complement and coagulation are affected by each other and how they turn on each other in the plasma. But first, I'm just going to tell you a little bit about that enzyme, that one that activates clotting factor 10. It consists of a transmembrane cofactor called tissue factor. It accelerates a plasma enzyme called factor 7A between 5 and 6 orders of magnitude. That is a huge accelerating event. These little dimples in my model indicate an enzyme active site. And together, factor 7A sitting down on a procoagulant phospholipid containing membrane, they activate clotting factor 10. Factor 10 can fall off as the factor 10A active enzyme can fall off, and they generate thrombin, abbreviated factor 2A. Factor 2A or thrombin can then directly initiate the polymerization of fibrin that makes the molecular mesh of the clot. Thrombin, as well as the nascent factor 10A, still within this tissue factor 7A complex, can then do an incredible bunch of things as initiators of cell modulation through protease activated receptors. And that's where this fits into so many different pathological and physiological mechanisms. Clotting also interacts and crosstalks in so many ways with complement, and that's what I mentioned that Alex Leatherdale is going to tell you about. And with that little tiny introduction, our primary foci of this talk is going to be clotting factor 10A and tissue factor and how they fit into clot busting and virus infection. And now I'm going to hand off to Alex Witt, who's going to tell you about clot busting. So Alex, take it away.
Thanks, Ed. To get blood clot formation, we need to generate fibrin, the mesh scaffolding of our clots. For that, we need the prothrombinase complex of factor 10A, factor 5A, and anionic phospholipids in green to cleave prothrombin into thrombin, the protease that can cleave fibrinogen to generate fibrin. Intact fibrin then acts as the meshwork of a clot until enough plasmin is generated to break it up. What we're going to highlight here is fibrinolysis, the process by which fibrin is cleaved by plasmin to dissolve the clot. When fibrin is intact in the scaffolding, it acts as a slow cofactor to bring together tissue plasminogen activator and plasminogen. Close proximity allows TPA to cleave the zymogen plasminogen into the active form plasmin, and plasmin is then able to begin clot busting by breaking up the fibrin. Plasmin's first cleavage of fibrin generates the C-terminal lysine here, a site at which plasmin preferentially cleaves fibrin in its next cleavage. Factor 10A in our prothrombinase complex here is the star of our talk today because it is the cofactor that requires binding to anionic phospholipids, localizing our clot to the site of vascular damage. Plasmin feeds back and cleaves 10A even quicker than it cleaves fibrin, generating a cofactor for TPA that can speed up this entire process. With TPA's role in clot degradation in mind, clinicians have been using recombinant variations of TPA for nearly 35 years as a thrombolytic agent, breaking down clots involved in serious medical conditions such as heart attacks and strokes. Alteplase, the FDA-approved thrombolytic, has been a huge clinical success, and it's enabled rapid research advances. However, an extremely high dose of TPA is required clinically and can actually result in severe hemorrhage bleeding in as many as 6% of patients. Most studies are conducted with high concentrations of TPA to mimic the clinical levels. However, high concentrations actually overcome our threshold for the slow cofactor effects because TPA can cleave plasminogen in the absence of a cofactor if the concentration is high enough. In this way, we didn't know much about the initiation steps of fibrinolysis because we were bypassing those slow cofactor thresholds. In our lab studies, we have used lower concentrations of TPA to focus on the initiating events of fibrinolysis and the possibility of auxiliary cofactors. Where we used to think that fibrin was the star of the show, we've come to realize that factor 10A can play a large role as well, and that's why it will be the focus of today's talk. Next slide, please, Ed. In an effort to generate a safer thrombolytic agent, our lab has investigated clotting factor 10A, which has impressive cofactor activity as it enhances the generation of plasmin by TPA in its cleaved form, 10A3313. Both factor 10A and 10A3313, originating from autoproteolysis or cleavage by plasmin, have been shown in this chromogenic assay to substantially increase the concentration of plasminogen-derived plasmin when compared to TPA alone. On the right-hand side here, we're looking at clotting factor 10A and its fragmentation process. From 10A alpha, we get 10A beta, which has an additional C-terminal lysine. And then in as little as 2 minutes, we can see the generation of 10A33, which has a second C-terminal lysine. If you'll recall, those C-terminal lysines make factor 10 even more attractive for plasminogen binding, and it makes factor 10A a localized TPA accelerator. So if you can see where we're going with this, factor 10A holds incredible promise as a thrombolytic agent. Next slide. If we come back to the idea of the localization, which for factor 10A means binding to anionic phospholipids, notice that when factor 10A is not bound to these phospholipids, we actually have a different set of cleavages on the left here. These cleavages do not generate the C-terminal lysines that facilitate new plasminogen binding sites. And as a consequence, 10A does not become a TPA cofactor and accelerator. When 10A is localized to the site of the clot, we do see the cleavage process such that C-terminal lysines are generated, and we get that 10A3313 species at the bottom that I mentioned earlier. In plasma, however, that second cleavage site renders a very, very short-lived species of 10A3313, which is very quickly cleaved into a 28 kilodalton species with no fibrinolytic activity whatsoever. In order to use factor 10A clinically, we need to prevent that last cleavage. Now, we also need to block the factor 10A active site. So unfortunately, it is called clotting factor 10A for a reason. And if we want to use this as a thrombolytic agent, we need to block its primary active site. So our lab has accomplished these two goals simultaneously in a molecule we call 10AIK. Through chloral methyl ketone chemical modification of the active site, we block the histidine of the active site and also prevent the cleavage of 10AIK into the 28 kilodalton species. You'll also note that we've added a C-terminal lysine to the end of our blocking agent, which enables our 10AIK derivative to hold the same number of C-terminal lysines as intrinsic 10A3313. This makes our modified 10AIK, a strong candidate for thrombolytic therapy. Unfortunately, as Ed mentioned, I've only recently arrived on scene in the Pryzdial Lab, so I will leave it up to Scott to show you how 10AIK acts in plasma.
Hey, thanks, Alex. As Alex mentioned, TPA is currently the drug of choice for treating thrombosis. Unfortunately, it comes with risk, and some patients experience a serious hemorrhage because TPA isn't targeted to the site of the thrombus. With this in mind, it became our goal to replace or reduce the amount of TPA required to effectively break up pathological clots. In 10AIK, we've created a targeted, enzymatically inactive form of 10A that has two C-terminal lysine residues available for interaction with TPA and plasminogen. This was accomplished, as Alex mentioned, by blocking the active site of factor 10A with a small molecule inhibitor that we had synthesized by the CDRD here at UBC. Attached to the inhibitor is a PEG spacer made up of 8 units of ethylene glycol and terminating in a lysine residue. This, combined with the intrinsic C-terminal lysine and membrane-binding GLA-domain, gives us a molecule that should be able to bind 2 plasminogen molecules and act as a super cofactor for TPA at the site of a clot. And it does this all without introducing any 10A clotting activity. This is a recipe for a perfect clot buster, and I'm excited to show you how it compares to TPA in vivo. We put 10AIK to the test in a variety of assays, including an occlusive thrombosis model in mice. Here we expose the carotid artery in a live, but anesthetized mouse, and attach a tiny ultrasound probe. The probe measures blood flow in real time as we generate a clot using ferric chloride, and then attempt to break up the clot by injecting the TPA variant tenecteplase, or our 10AIK, into the tail vein. This is a well-established model that we brought from Bill Sheffield's lab in Hamilton to the MBF at UBC. Next slide. Here you can see what it looks like after the occlusive clot has been generated. The clot is made by placing a piece of ferric chloride soaked filter paper directly onto the carotid artery for a defined period of time, and then removing it and washing away the excess ferric chloride. It's actually super cool to watch this. You can see the clot form with little bits breaking off and then reforming to finally fully occlude in about 7 minutes. Once we have this stable clot, drugs can be injected into the tail vein to try to break it up. And then down below, you can see that the clot has been fully lysed and blood can flow freely again. And this flow is measured over the course of a 1-hour experiment and the area under the curve is calculated for flow over time. On the right, you can see that in the absence of a drug, very little flow is recorded since the carotid is occluded the whole time. If we inject a therapeutic dose of TPA, however, it does its job nicely and busts the clot, giving a high amount of flow. Next, in line with our goal, we found that the effective dose of TPA could be cut in half with the addition of our 10-AIK. This opens up the possibility for an adjunctive therapy and a drug eventually. Finally, and excitingly, even an injection of 10-AIK alone restores blood flow just as well as the TPA, and it's dose dependent. So these results look really great, and it was clear that our clots were being thoroughly lysed by both TPA and 10-AIK. But one has to wonder if we're also causing bleeding at the same time. To investigate this possibility, we looked at surrogate markers of systemic fibrinolysis by Western blot. These here are Western blots of mouse plasma collected at the end of the carotid ultrasound experiments. Each lane represents one mouse in the study, and we're using fibrinogen degradation products and the generation of plasmin from plasminogen as markers for systemic fibrinolysis. Once again, you can see that in the absence of either drug, there are only the expected fibrinogen chains lighting up on the blot, and there's no plasmin visible at all. Injection of the therapeutic dose of TPA generates all these fragments here of fibrinogen and enough plasmin to detect by blot. We were thrilled and relieved to see none of this resulting from even a double dose of 10-AIK, despite successful reperfusion in the carotid ultrasound. These results indicate that while TPA is activating the fibrinolysis system on a large scale, 10-AIK seems to be acting primarily on the thrombus and not causing systemic lysis. Now, further to these surrogate biomarkers, we recently embarked on a collaboration with the Multifunctional Lab in the Center for Brain Health to look at an intracranial model of hemorrhage. In this experiment, we're looking for bleeding into the brain resulting from clot busting treatment. To do this, mice were first injected with Evans blue dye, which binds to albumin in the blood. They were then injected with either TPA or 10-AIK, which was allowed to circulate for 1 hour. The mice were then perfused to remove all blood from the vessels, and the brains were collected. Evans blue fluorescence in the brain tissue could then be quantified, showing leakage from the vessels into the brain. Now, these are just preliminary data, but you can see with just a little bit of squinting that TPA produces more signal than 10-AIK. When quantified, there's a clear difference observed in both the dorsal and ventral sections of the brain. This is really encouraging, but there's still a lot more work to be done, which is ongoing now. Next slide, please. I hope that I've convinced you that modified factor 10A shows promise as a clot-busting therapeutic. 10-AIK is a great starting point, but it still has some drawbacks. The precursor, factor 10, is plasma-derived, which brings concern of infectious disease. It also has to be purified by a multi-step process, and it must be enzymatically converted to 10A. The purified 10A then has to be chemically modified to tether a C-terminal lysine.
Yes,
And finally, the clotting active site of factor 10A must be completely blocked. This is a lengthy and unappealing process for a drug company, but these are all challenges that can be eliminated by the production of a recombinant factor 10, lacking enzymatic activity and modified to present a C-terminal lysine residue. A panel of mutants has been created to do exactly this and will be tested in our mouse models very soon. And now we'll switch gears a little bit and pass it on to Michael.
Michael, before we start, I'm just going to ask people in the audience to kindly turn off your microphone.
Awesome. Thanks, Scott. Thanks, Ed. Hi, I'm Michael. I'm a research associate in Ed's lab. And like he spoiled a little bit before, I've been with them for over 25 years. It's actually 27 years in September. And to my credit, I've only been working on the virus project for 26 years. So it's really, really new. Like Scott said, we're switching gears a little bit. And we'll be talking about the virus projects that are ongoing in the lab that are trying to determine an underlying basis between infection and chronic diseases. And why we're interested in this is that numerous virus types dysregulate the clotting system and correlate to pathology. The resulting diseases range widely depending on the virus and are driven by three mechanisms. The first are hemostatic proteins, which Henry and I will talk about. The second are circulating blood cells, which is today's project, who's unfortunately unable to be here today. And the third is interactions with the complement system, which Alex Leatherdale will be finishing off our seminar with today. And emphasizing the prevalence of abnormal clotting in viral pathology, herpes simplex virus type 1, HIV, dengue virus, influenza, Ebola, and SARS infection have all been associated with alterations in clotting. To explain the diverse list, a common molecular basis is speculated. Virus envelope constituents are the first to contact host hemostatic factors and govern infection of cells. Yet, roles of host-encoded proteins exploited on the virus surface have been under-investigated. To fill this knowledge gap, our initial studies have been on herpes simplex virus, simply shortened to HSV in my slides, or simply virus on the slides, which for decades has been linked to vascular disease, including a clear cause and effect relationship in several animal models. Speculating that coagulation may be advantageous to the virus, initial experiments were conducted using highly purified HSV particles and thrombin to determine the effect on infectivity in a standard plaque assay. And this is depicted on the left side of the slide, where the large circular structure is our representation of a virus particle. And as I'd mentioned before, the orange circle there is thrombin. The results from this assay are seen on the right-hand side of the slide, in which we determine infectious events following staining of a cell monolayer. These can be seen as white circles in the top panel when virus was added alone. However, when thrombin was added with the virus, as seen in the lower panel, there was a noticeable increase in infection. And this gave an indication that coagulation proteases are important to virus infection. Next slide, please. So tissue factor, and I'm going to refer to that as TF on my slides and as I give my presentation, is a transmembrane cofactor essential for mammalian life. And as mentioned earlier by Ed, accessible tissue factor triggers blood clotting by accelerating factor 7A mediated proteolytic activation of factor 10 to factor 10A. To interact effectively, these clotting factors also require the availability of anionic phospholipid containing membranes, which like TF is stimulus induced in order to localize the response. Importantly, our earlier work many decades ago showed that both TF and anionic phospholipid were functionally available on the surface of several members of the herpes virus family, thus bypassing the strict cellular control. We hypothesized that since TF bridges hemostasis and inflammation, virus envelope tissue factor may play a role at several stages of infection and other chronic diseases. To get this project started, we wanted to delineate the function of TF on the virus surface using a unique panel of viruses. As shown on the left side of this slide, in the experimental flowchart, HSV type 1 was grown initially in a tissue factor deficient cell line. Since the virus acquires tissue factor from the cell in which it replicates, the resulting progeny virus was also TF deficient. A TF inducible cell line was created from the original, and once infected, generated TF containing virus particles shown on the right of the flowchart. The matched pair of TF characterized cells provided the opportunity to limit the differences in host acquired constituents on the virus surface to tissue factor and provided a means to specifically evaluate the role of TF in the infection process. To determine if viral TF could substitute for its cellular counterpoint, the cofactor involvement of tissue factor with 3 different coagulation proteases in HSV infection was investigated. Here we show that infection is increased with each of the 3 proteases, when each of the 3 proteases are included with both the tissue factor positive and tissue factor negative viruses, as seen in the IgG lanes in the right-hand panel. However, an anti-tissue factor antibody, which is denoted as anti-TF, known to attenuate the signaling function of tissue factor was added. The enhancement of TF positive HSV1 infection by factor 10 or 7A was lost. The reduced HSV infection is consistent with 7A and 10A being organized within tissue factor on the virus surface, analogous to the tissue factor complex mentioned earlier. Importantly, the tissue factor antibody had no effect on the thrombin-dependent enhanced infection using the tissue factor positive HSV1. In sharp contrast to the tissue factor positive virus, the tissue factor antibody lost its significant effect on factor 10A and factor 7A mediated enhancement of infection. These data demonstrated that tissue factor associated with the virus and not the target cell was responsible for the presentation of factor 10A and 7A to the cell surface receptors, and thus promoting infection. Next slide, please. Okay, coagulation proteases transduce intracellular changes such as kinase activity, gene transcription, and protein synthesis through the activation of protease activator receptors, known as PARs, on the cell surface. So PAR type 1 has been well characterized and is associated primarily with activated primarily by thrombin, in addition to its clotting factor, tissue factor participates in cell signaling through the formation of tissue factor 7A 10A complex via PAR2. The demonstration that viral TF functions analogous to cell surface TF in assembling coagulation protease complexes highlighted the importance of host-derived envelope factors in the infection cycle. The role of PARs in thrombin, 10A, and 7A effects on the tissue factor positive virus infection was further evaluated with cleavage blocking antibodies shown as anti-PARs on the right-hand panel. As shown on the right, the anti-PAR1 antibody specifically inhibited the tissue factor dependent increase in infection. However, anti-PAR1 had no effect on the enhancement of infection caused by either 10A or 7A. In contrast, a blocking antibody to PAR2 inhibited the 10A or 7A dependent enhancement of virus infection without affecting thrombin supported infection. The results presented here show that PAR1 and PAR2 mediate distinct effects on viral infectivity. And while thrombin promotes infection dependent on PAR1, that is independent of viral TF, optimal 7A and 10A enhanced virus infection is remarkably dependent on tissue factor and facilitated by PAR2. Next slide, please. Okay, the potential effect of virus surface TF on infection in vivo was evaluated by comparing HSV1 positive and HSV1 negative virus infection in Balbsy mice, as this strain is highly permissive to infection. Mice were inoculated with a sublethal dose of either tissue factor positive or tissue factor negative virus by intravenous tail injection. 3 days post-infection, 5 organs were recovered and evaluated for virus infiltration, including the lung, heart, spinal cord, brain, and liver. HSV1 infectivity is shown in the panel on the right, and viral genome, which isn't shown, per mg of tissue were quantified. Each organ was shown to accumulate infectious virus, and infectivity was found to be highly dependent on the presence of envelope TF. Data not shown further demonstrated the role of tissue factor in vivo when a combination of tissue factor antibodies that recognize only the TF incorporated into the purified virus envelope had reduced infection. Therefore, the physiological initiator of coagulation, tissue factor, associated with the HSV1 envelope is a previously unrecognized host cell-derived constituent that serves an important function during the in vivo virus infection cycle. Next slide, please. The involvement of tissue factor in 10A production and the consequent downstream generation of thrombin and effects on HSV1 infection in vivo were investigated using specific coagulation protease inhibitors. Since infection by TF negative virus was very low, only infection by TF positive HSV1 was evaluated. Here we show that all 3 anticoagulants studied blocked infection in each of the organs studied. Thrombin is the ultimate coagulation protease and is produced once TF mediates 10A generation. To investigate the possibility that thrombin has a role in the virus life cycle, infection was measured in the presence of a highly specific thrombin inhibitor, herudin. Indeed, herudin attenuated in vivo infection, indicating that virus exploits thrombin activity for infection. However, Inhibition of infection was also facilitated by NAPC2, a known inhibitor of 10A, and the tissue factor 7A10A complex, suggesting that upstream factor 10A may also be involved. Therefore, the effect of a 10A-directed anticoagulant was investigated. At the approximate therapeutic dose used in humans, apixaban, which is a widely used small molecule 10A-directed anticoagulant therapeutic, was as effective as NAPC2 at reducing HSV-1 infection, suggesting that inhibition of 10A may be the dominant antiviral effector. So these data suggest an unforeseen application of apixaban or other routinely used anticoagulant therapeutics as antivirals. Next slide, please, Ed. Okay, so following the discovery of all this with the HSV-1, enhanced infection by tissue factor on the surface, and this was efficiently inhibited by anticoagulants in vivo, we wanted to test the direct tissue factor pathway anticoagulants on SARS-CoV-2, as SARS-CoV-2 antiviral agents, since SARS-CoV-2 also initiates profound dysregulation of coagulation and inflammation. This will be tested in a humanized bone marrow liver thymus mouse model that is further modified by engrafting human lung tissue. These mice are therefore reconstituted with the human innate and adaptive immune systems and are capable of an appropriate immune response against the virus, allowing by the transplanted human tissue. To initiate our leaps into the coronavirus world, our coronavirus experiments were initiated with SARS-CoV-2 seen on the right-hand side, the left-hand side, as this virus has a lower biohazard compared to SARS-CoV-2. 229E shown here in the EM picture has a characteristic corona of spike proteins surrounding the virus. In order to go to the next stage, we first need to propagate and purify the SARS-CoV-2 virus in the UBC CL3 Facility for Infectious Disease and Epidemic Research, which is also known as FINDER. This has proven to be a challenge both operationally in terms of accessing the facility and logistically with respects to executing procedures within the space. However, we have persevered, and within FINDER, I've been able to generate infectious SARS-CoV-2 as seen in the virus growth curve on the right, with a peak in virus production by day 3 post-infection. Additionally, in preliminary experiments using virus-infected media, I was able to show a serum-dependent increase in infection similar to what we've seen for HSV-1, suggesting a common antiviral target. And with that, I'm going to switch over to Henry.
Thank you, Michael. So yeah, I'm Henry. I'm a first-year graduate student with the Pryzdial Lab, though I have been with the lab as an undergraduate since 2019. And so far, you've seen some of the results of tissue factor activity on the surface of HSV-1. But beyond this, tissue factor has been observed on the surface of every single envelope virus that the lab has investigated thus far. And we do this by a technique called immunogold electron microscopy, where antibodies are conjugated to gold nanoparticles of a known size. So when we image the virus, the bead appears as a round black dot, as you can see in these electron micrographs to the right, which have been captured by Dr. Brian Lin, who's a recent graduate from the Pryzdial Lab. And here, the anti-tissue factor antibody is conjugated to the larger gold beads in these images, and the much smaller, tiny gold beads are conjugated to antibodies against specific viral proteins. As we expect, based on what Michael has been showing, this propagated HSV-1 variant stains for both tissue factor and a HSV-1 envelope protein. Another envelope virus that's commonly studied in our lab, especially by today, who's here, which is dengue virus, also co-stains positive for tissue factor and the dengue virus-specific envelope protein. And we have also observed this tissue factor appearance on virus particles from infected patient plasma, ones that have never touched a cell before, besides human cells. These HIV particles shown here show both tissue factor antigen and the HIV envelope protein. And these hepatitis C virus particles also co-stain positive for tissue factor and surface glycoprotein. And we believe that this tissue factor exposure on the surface of these viruses is near ubiquitous. And as we previously mentioned, viral infection can affect coagulation in a number of ways, but our lab hypothesizes that this tissue factor on the surface of viruses can directly contribute to coagulation. Next slide, please. So one of the ways that we measure this is a plasma clotting assay where the virus is added to plasma, which is recalcified to initiate coagulation. And here the only source of tissue factor and anionic phospholipid, which are necessary for the formation of 10As and then further a clot in this system, is the virus, meaning that the more virus we add, the faster the plasma should clot. From this left data panel here, we see a titratable effect on clotting times as dengue virus concentration increases, with a decreased clotting time seen with higher dengue virus concentration. And so the same inhibitors that Michael has mentioned were employed here as well, with NAPC2 lengthening the plasma clotting time to the cutoff for this experiment, which is 300 seconds in our lab. The anti-tissue factor antibody also increased the clotting time, though not to the same extent as NAPC2. Next slide, please. Thank you. So another method that we use to assess the procoagulant activity of tissue factor on the surface of these viruses is a purified factor 10A generation assay. And the components of 10As are mixed together. And as before, the only source of tissue factor and anionic phospholipid in this experiment is the virus. Generation of 10A follows through the cleavage of a chromogenic substrate with more factor 10A generation, meaning more tissue factor activity. So as we get more virus, we should expect a greater amount of 10A generated. And in the data panel on the right, we do see a decrease in factor 10A generation as we add less dengue virus to the reaction. And these same inhibitors, NAPC2 and the anti-tissue factor antibody, are employed in this experiment as well. And these greatly reduce the amount of 10A generation, even when they are combined with the highest concentration of virus. But based on these data and similar experiments conducted on other viruses, including HIV, we believe that this viral tissue factor could be a contributing factor to the procoagulant pathology seen in viral infection. Next slide, please. Switching gears a bit, we are also investigating other ways that viral infection can result in these coagulopathies. And aberrant coagulation is common in COVID-19. And there are also a large variety of upregulated pro-inflammatory markers in these COVID-19 patients' blood. And many of these are known to induce tissue factor expression and activity on endothelial cells. So we are in the process of developing an assay where COVID-19 patient sera or sera from healthy controls is used to pre-treat an endothelial cell monolayer, which is then washed off and we replace it with plasma. And then thrombin generation is followed with the cleavage of a fluorogenic substrate. If we look at the data panel on the right, the black and blue curves in the middle show cells that are pre-treated with healthy control sera or the bucket that was used. But the red curve shows thrombin generation on cells pre-treated with COVID-19 patient plasma, with both a greater total amount of thrombin generation and a steeper slope, showing faster thrombin generation as opposed to the cells treated with normal control sera. And interestingly, blocking tissue factor activity with an antibody on these cells pre-treated with COVID-19 sera, which is shown in the green curve, results in no thrombin generation, showing that this procoagulant activity that is displayed by these cells is initiated by tissue factor. Next slide, please. So a component of our innate immune system, the complement cascade, has a number of known and certain proposed links with the coagulation cascade. And two of these proposed links are shown here with C5b, C6, and C7 complex, resulting in the activation of procoagulant tissue factor activity by decryption, or a C5b, C9 complex or deposition on the cells, resulting in anionic phospholipid exposure. Given the potential and known links between complement and coagulation, Alex is going to tell you a bit more about the relationship between COVID-19 and complement, and also the potential of using these activated complement proteins as biomarkers in COVID-19 patients.
Thanks, Henry. So I'll start with a brief overview of COVID and the complement system. The component of the innate immune system known as complement has been implicated as a driving factor in both the progression and severity of COVID-19. And the mechanisms through which complement is activated by SARS-CoV-2 are unclear, but there are some studies that have found elevated complement activation markers in the blood of COVID-19 patients that increase with disease severity. There's also large amounts of activated complement proteins found in the lungs, kidneys, and liver of patients who have died from COVID-19. And in vitro studies so far have demonstrated that the closely related SARS-CoV nucleocapsid protein can bind to MASP2, triggering complement activation through the lectin pathway. And lastly, the SARS-CoV-2 spike protein has been shown to activate the alternative pathway, potentially by inhibiting regulation of factor H. Next slide, please. So as a review, the complement system is a proteolytic cascade that acts as a first line of defense against injury and infection to neutralize pathogens and alert and recruit immune cells. Complement can be activated through three interrelated pathways, the classical, lectin, and alternative. These pathways converge at the formation of C5 convertases, which cleave C5 into C5a and C5b. C5a is an anaphylotoxin that triggers local degranulation and leukocyte chemotaxis, and C5b binds to the cell membrane, acting as the nucleus for formation of the membrane attack complex, a multi-protein complex that inserts into the cell membrane with the goal of killing the targeted cell. And both infected or damaged host cells can become the target of complement, leading to widespread organ damage when it is pathologically active. Next slide, please. There are many, back one, please. There are many known crosstalk mechanisms between complement and coagulation, suggesting that the innate immune response to infection is inextricably linked to the activation of coagulation. And one of our ongoing projects has been to characterize the role of complement in the pathogenesis of COVID-19 related coagulopathies. Next slide. So far, we have found that there is a striking increase in serum markers of terminal pathway activation, C5a, and the soluble form of the membrane attack complex, SC5b9. So at the time of ICU admission, critically ill COVID-19 patients have a 9-fold median increase in C5a and a 5-fold median increase in SC5b9 compared to healthy controls. And given the feedback mechanisms between coagulation and complement, we also assayed C5a and SC5b9 in autologous EDTA plasma and found similar effect sizes in those samples as well. Next slide. The alternative pathway activation products, the BA fragment and factor BB, were significantly elevated in patients compared to controls at the time of ICU admission as well. Next slide. We found that a subset of patients had elevated factor D, which is the rate-limiting enzyme of the alternative pathway, which likely contributes to the magnitude of alternative pathway activation in some individuals. Conversely, the negative regulator of the alternative pathway, factor H, was significantly lower in patients compared to controls, although the cause of this difference so far is unclear. Next slide. When we correlated circulating complement analytes to respiratory status on the same day that the blood samples were taken, we found that alternative pathway analytes BB and BA and terminal pathway analytes C5a were significantly associated with the severity of hypoxemia, similar to what we had previously observed in IL-6. Next slide. Next, we examined complement activation longitudinally in a subset of patients that had multiple blood samples available. We found that both the alternative pathway and terminal pathway markers remained elevated over the entire 21-day study period. These values were well above the upper limit of the 95% confidence interval of the control cohort. And interestingly, complement appeared to fluctuate in response to corticosteroid treatment. Next slide. So in light of their persistent elevation throughout hospitalization, we found that the concentrations of the BA fragment and factor D at the time of ICU admission showed utility in predicting mortality in the following 30 days. The odds of dying increased 4-fold for every 1 unit increase in BA or factor D on average in this cohort. These values were comparable to other known risk factors, such as obesity and diabetes. Whereas D-dimer, which has been proposed as a robust predictor of mortality in various other cohort studies, was a poor determinant of survivorship within our cohort of critically ill patients. So complement status may therefore be more sensitive to predict deaths in patients who are already extremely sick, whereas other proposed markers tend to be derived from cohorts composed of people with a range of severities, from mild to severe. And with that, I will hand it back to Ed Pryzdial.
Thanks very much, Alex. Well, like I said, clotting factors are multifunctional. We talked about pathology. We also talked about physiology, probably unknown to you because we are hypothesizing that 10A is involved in the physiological process of fibrinolysis, and that's where we're deriving our new clot busting therapeutic for treatment in thrombosis. That being factor 10A and 10AIK as a new paradigm for thrombolytic therapy development. We talked about tissue factor existing on the surface of enveloped viruses, and all of the ones that we've looked at to date seem to have this. We talked about viral tissue factor inducing clot formation and enhancing infection, at least in one virus we have looked at in the infection process, and we're now looking at others. We also talked about virus tissue factor providing a broad specificity antiviral target because it seems to be ubiquitous on the viruses that we have all looked at today. COVID-19 serum induces endothelial tissue factor, explaining the thrombotic microangiopathy that is prevalent in the literature and more importantly, prevalent in patients that get COVID-19. And as Alex just told you, complement is a very strong biomarker for hypoxemia and mortality in COVID-19. This is our gang, and we're sorry that Xai-K could not join us today to tell you about her proteomics and how dengue virus affects platelets. And we are grateful for our fibrinolysis collaborators, Bill Sheffield and Brian McVicker, and Louis-Philippe Bernier did those brain scans that Scott showed you today. And we would be also lost without our virus collaborators, and especially Ed Conway, who is unable to be with us, I think, today, Mark Horowitz, and Ivan Sadowski from UBC, and so many others have been instrumental in our studies. So with that, I'd be happy, and we'd be happy to take questions. Thanks very much.